That was fantastic.
Billy, I think we have a question here.
We do? Okay.
Instead of diet modification / increased food diversity, can some of the same gains be made by adjusting prebiotics and probiotics? Do you see this as a potential therapy going forward?
Sure. I think that it can. We have a lot of work to do. I think that, nutrition may be almost, and this is taking into account that I understand that nutritional intake can be a real problem in these kids or adults. But nutrition may not affect just one bug. It may affect 20 or 30 different bugs or 3,000 bugs. And we're trying to figure that out. So I think a lot of it is going to depend on how many bugs are affected, who they're affected in. I think that a more reasonable assessment may be that we may be able to have, say, pre or pro, or now postbiotics are a newer thing or emerging too, that may target specific traits that are associated or specific behaviors or specific gut issues rather than tackling everything at one time. And that's why I talked a little bit more about this personalized approach.
Great. Billy, you had a question?
Yes. So, and I think this is a question for both of you. So one of the goals that we have is we'd like to help inform future program announcements and RFAs from funding agencies about what the community of both families and scientists want money to be spent on. And I'm wondering if both of you could comment on what guidance would you give program officers and others writing program announcements? How should we be spending resources, and what general areas should we really focus on to move all of this forward?
Sure. Go ahead, Kay.
Okay, so I think you have to think in terms of what your goals are. Do you want to do the basic approach, or do you want to do, which really leads you into future long-term thinking, or do you need to do something here and now while everyone is looking for longer term curative type results? Much of the work that I have done up to this point has been the here and now. What can we do to help our families improve the quality of life of their children here and now? Because so much of the curative aspects involve basic work that will take a much longer time period. So I would say the first thing you do is sort out where are your priorities for the monies that you have. Here and now, more appropriate clinical work versus longer term avenues, such as, for example, the microbiome. If I weren't so old, the microbiome would be near and dear to my heart as well. We've done some of the studies in Rett syndrome as well. And one of the things that can certainly be expanded upon is looking at the metabolome as well. In our girls with Rett syndrome, we have some very distinct differences between affected and unaffected individuals when we look at the amino acid type metabolites or the precursors of the neurotransmitters. And I think that's an avenue also that needs to be explored for the long-term consequences. Dr. Margolis didn't talk about the serotonin aspects of the gut microbiome, but she knows as well as many of us know, there are many neurotransmitters that are different in affected individuals compared with unaffected individuals. And so how you can interrelate one diet to the microbiome, and 3, the metabolome, and then 4, the behaviors, is a very complex analysis that requires numbers of children in a particular study. And so I think you do have to think about multi-center studies as well, as Dr. Margolis has done. I think that's an excellent outcome with so many people that have been included in her studies, which is very difficult to do. It sounds like it should be quite simple to collect a stool sample, but it can be difficult because parents have many things on their hands, including the care of their children.
And I actually can vouch for that. My daughter was a participant in that study through Baylor. And we got all the materials thinking, oh, this will be really easy. And it was more of an ordeal. I was like, yeah, I'm a poop doctor. I can collect poop. That's a really good point. And I think it speaks to the fact of making sure that we're being inclusive in research. And we typically approach research as how we would in a typical population, as far as the implementation side of things. So I'm glad to hear you mention that.
Well, and certainly, when you have an engaged audience, and that is so, when our families recognize what we're doing and why we are doing it and what we need, many of them really do come forth, and it's commendable with all the things that you have to do to take care of a child with special needs. I think it's commendable when our parents are enthused, excited, and engaged in the work that we do. And we can't thank them enough because we really do need our families.
So I think just to build on what Kay just said, I mean, multi-center, different geography, different levels of autism, and family, and organizational input. I think that the advantage that the study I presented had is that Ruth Ann Luna is very vocal about the fact that her son has severe autism. And so having somebody in the planning stages that understood, and even with data analysis of what the limitations were and planning our prospective study, which is actually all kids with severe autism, it was so helpful to have her guidance in terms of what we could and could not do and what the reasonable expectations were for families who, I think a lot of times want to be active participants, but, really struggle. I think, we lack, so I think multi-center large studies are critical given the diversity. I think we miss a lot with neurodevelopmental disorders and the severe end of them because they're the hardest patients to get samples from. And so there's not a lot of studies looking at that. And when we actually try to quantify behavior severity or autism severity, there's really no great scales to objectively take that into account, which is, which can be a very challenging thing and would naturally exclude families that can't do that, or if we can't make it applicable, make it easy for them to participate. I think,
Dr. Margolis, I would challenge you to go ahead and develop your autism GI quality of life or gastrointestinal health questionnaire, which is what we basically had to do in Rett syndrome. And we're trying to expand that to other MECP2 disorders. But you are correct. The reality is there is a very broad spectrum within any specific disease entity or disorder entity. And so in order to, again, narrow down what may be affecting the outcome of your different parameters, having a tool that does look at the individuality and then the group as a whole in terms of the spectrum of the disorder would certainly help.
Sure. I think appreciating the complexity, and I think that's a relatively new concept, not amongst the families and not amongst the physicians that treat these families or work with them, but the idea that, neurodevelopmental disorders are not just the brain. There's many other medical issues, and they're not necessarily all at least known to be neuron or nervous system related. And so I think appreciating that and being able to separate that out is also going to be critical when studies are brought forth. Anybody that wants to just study autism as one label and promotes that and submits a grant like that, that should be knocked off the bat right away, because there's just, it's not, no disease state, as we're learning, is one disease state, and certainly neurodevelopmental disorders, as we know, are far from that.
I think the other thing that all of you can think about is having statistical input from a statistician who not only understands the math and the multiple complexity analyses that you need to use when you're dealing with something such as a microbiome, a behavioral input, a metabolome input, and then a clinical characteristic input. But the statistician or the statistical support system needs to be able to understand the biology as well as the mathematical components. And so often that's a very difficult person to find, because the statistical support entity is often not based on a multidisciplinary approach to helping research activities. So I think that's also a very useful consideration when you're thinking about how do you spend your money.
And just a plug for Calliope, who is actually really working towards developing these screening technologies that are also, building upon, what we published on, but going many steps further into really figuring out the right ways to screen children with neurodevelopmental disorders.
It's a strong argument for more and more collaboration. I think we're all, like, all of the stakeholders are strengthened when we do that. Heidi, I think we're going to move to the next session now.
Yes, and thank you both, Dr. Motil and Dr. Margolis. I think you've highlighted. Dr. Jimenez Gomez put up a slide that had limitations, and one of the things that's been a ongoing theme is the ability to measure meaningful change. So we'll continue to move forward working on that and hear about it tomorrow from Dr. Calliope Holling. And we have one more session. So thank you all for sticking with us.